Welcome back to the Cell and Molecular Biology course. I'm your coach instructor Nia Pendra. Welcome to this presentation on the discovery of the double helix structure of DNA. Today we will travel through time and explore key milestones that led to the revelation of DNA's iconic structure, a discovery that revolutionized modern biology and medicine. Our journey begins in 1869 when Swiss scientist Friedrich Miescher first isolated a mysterious substance from the nuclei of white blood cells. He called it as nuclein. At that time, its role was unknown, but this substance was actually DNA. The molecule, we know how it carries the genetic information. So this was the first discovery which led to the double helix structure of DNA. So this was the first milestone in the discovery of DNA. So fast forward to 1928, British bacteriologist Frederick Griffith, he performed experiments with pneumonia-causing bacteria in mice. So he observed something remarkable, that is dead virulent bacteria could transform harmless ones into killers. In the illustration, you can see the rough strain and smooth strain. The rough strain is non-virulent one with which the mouse survives, but when the smooth strain is injected, it dies, which is a virulent strain. So in the third step, you can see the heat-killed smooth strain, which when injected, the mouse still survives. But when the rough strain and the heat-killed smooth strain are together injected into the mouse dies. So this is the experiment done by Griffith. It's called transformation experiment. So this hinted at the existence of a genetic material, though it wasn't yet clearly known whether it's DNA or proteins which were responsible for this condition. So this discovery of transforming principle is the next part of the discovery of the double helix structure of DNA. Next, in 1944, Oswald Avery and his colleagues Colin MacLeod and Maclean McCarthy solved the puzzle. That is, they identified DNA as the transforming principle, which is responsible for Griffith's results. In Griffith's results, they did not conclude that if it's DNA or protein which is responsible for that transforming uh, process which is occurring. So in this experiment, Avery, MacLeod, and McCarthy, their findings suggested for the first time that DNA was the molecule of heredity, though the scientific community remained divided. They didn't accept it that it is DNA which is doing all these uh, process. So the scientific community didn't accept at that time that DNA is the one which is carrying the genes and it is responsible for all these principles. It is not accepted at that time when it was discovered. In the illustration, you can see the S prime extract, which is nothing but the smooth strain extract. It is divided and injected into mouse to see if what is the molecule which is responsible for this transformation of virulent strain. So, first, the polysaccharides were destroyed, and then the remaining is injected into the mouse, and the mouse dies, and the live string is recovered. And then in the second step, the lipid is destroyed, and the rest of it is injected to the mouse, and still the mouse dies. And then the string is, which is live, is recovered. And then similarly, RNA and proteins were destroyed, but still the mouse dies. So all these were not responsible for the transforming principle. And finally, the DNA was destroyed, and the rest of the extract were injected into the mouse, and the mouse survived, and no live extreme was recovered. So this showed that DNA is the transforming principle, and this is the first strong evidence that DNA carries genes. So this experiment was the first step in which it proved that DNA is the one which is responsible for the transforming principle. And next comes Chargaff's rules in 1950. We have seen this rule in the structure of DNA. So yet again, we will be seeing in this. Then came after the Avery MacLeod and McCarthy experiments. Then came Edwin Chargaff's important discovery in the 1950. He found that in DNA, the amount of adenine was always equal to thymine and guanine was always equal to cytosine. Their ratios, known as the Chargaff's rules, provided a crucial clue that the bases pair up in specific ways. So these findings hinted at the structural logic underlying the DNA. So this is the Chargaff rules. In 1952, Alfred Hershey and Martha Chase performed their famous blender experiment using viruses called bacteriophages. Bacteriophages are nothing but the viruses which affect bacteria. So they demonstrated conclusively that DNA and not proteins was the genetic material which is passed from one generation to the next. So this was a pivotal moment, shifting the focus squarely onto the DNA. So in the illustration, you can see sulfur-labeled protein capsule, which is red in color. It is then injected. This bacteriophage is allowed to affect the bacteria, and then inside it, it is releasing the DNA, and the capsids are released. After centrifugation, no sulfur was found in the cells. So this showed that the protein capsule is not the genetic material. And then in the other experiment, they did in the other step, they are using the phosphorus labeled DNA, which is green in color. And then when this bacteriophages affected the bacteria, they released the DNA or the genetic material into the cell, and it after infection, and then after infection, these cells are blended, and then the centrifugation we can find phosphorus in the cells. So this was the experiment, which is Hershey-Chase experiment, which found that DNA and not the protein is the genetic material, which was confirmed using bacteriophages. So this was the next step in the double helix discovery. That same year, Rosalind Franklin used X-ray diffraction techniques to take a picture that would become legendary Photo 51. This image reveals that DNA has a helical structure Although Franklin's data was shared without her consent, it provided the missing piece that guided others to the correct model. So, Rosalind Franklin played a crucial role in the discovery of double helix structure of DNA. Her meticulous work provided clear evidence of the helical form and the dimensions of the helix. Unfortunately, her data was shared with uh, Watson and Crick without her permission, which helped them finalize their model. And today, Franklin is rightly recognized as a key figure in the discovery of DNA structure, and her contributions are honored worldwide. But Nobel Prize it was not awarded to her as she was not alive at that time. In 1953, James Watson and Francis Crick built up Franklin's X-ray images and Chertoff's rules, and they proposed the double helix model of DNA with two strands wound around each other, with bases pad in the center, and their model elegantly explained how genetic information is copied and it is passed on from one generation to the other. So their famous paper appeared in Nature in April 1953. So this was known as Watson and Crick's double helix model, and in 1962, Watson Crick and Maurice Wilkins were awarded the Nobel Prize in Physiology or Medicine for their discovery. But sadly, Rosalind Franklin had passed away in 1958 and was not eligible, though her vital contribution is now widely acknowledged. The discovery of DNA's double helix structure was not the achievement of one person, but the uh, culmination of nearly a century of work by many brilliant minds. So, this discovery laid the foundation for modern genetics, leading ways to advances like genetic engineering, forensic science, and personalized medicine. So, understanding the structure of DNA unlocked the secrets of life itself. Thank you for watching. I hope this timeline helped you appreciate the incredible journey behind one of the science's greatest breakthrough. See you in the next video.